Everyone's at home quarantine, so I assume it was easy to do. All right, so I'm going to talk about my Teps research. So this is a combination of two projects, effectively, about the origins and habitability of liquid water on ancient Mars. So part one is going to be about how you can get liquid water on Mars, and part two is going to be, assuming you have this liquid water now, is it habitable? All right, so let's see. All right, part one. So liquid water on Mars. So first of all, it's why do we think there was liquid water on Mars? And that has to do, so I'm talking specifically here about ancient Mars. And that looks at, there's a couple arguments for believing that there was large amount of water on Mars at one point in time. And there's two main arguments for this. One is geomorphology, which is just looking at surface features of Mars. They look very similar to terrestrial features that we know are caused by flowing water. So for example, our valley networks, craigs, crater lakes, shorelines. So these images here, the two left-hand panels are actually images of Earth, and the two right-hand panels are images of Mars, and showing very similar types of features. You can see canyons, you see these little river networks that you see on Mars as well. So that's one argument is, the surface looks like there was at one point in time flowing water. The other main argument comes from geochemistry, which is sort of what we see on the surface. So we see a lot of phyllosilicates, which are something that would be formed in the presence of warm and wet environments. The chemistry required to produce those needs certain temperatures and aqueous conditions. There's a lack of carbonates in some places, and those are stuff that you would want in a warm and dry environment to form. So there are some, but not a lot of them. So that kind of leads again to this probability that you may have had a wet environment. And there's a couple other arguments, a couple other geochemical cases for this, that you would have water at some point on the surface. But this is sort of at odds with the atmosphere of Mars. So if you want to have liquid water on the surface and be warm and wet, as we like to call it, you need to have a certain surface temperature. In order to get a certain surface temperature, you need an atmosphere to produce a strong enough greenhouse effect to warm the surface of the planet. Now, this is a problem because, at least in present day, Mars has no atmosphere. So if you were to extrapolate back in time, assume that it had an atmosphere that it once lost, most models don't produce a strong enough atmosphere to warm the surface. And there's a couple reasons for that. One of them is, eons ago, the sun was not as strong as it was today. So you don't have as much incoming solar energy to warm the surface. The atmospheric modeling for total atmospheric pressure in the past puts an atmosphere that's probably between 1 and 2 bar, which is fairly low in order to have enough gas to provide sufficient trapping. And the other problem is sort of the nature of that atmosphere. CO2 alone does not provide enough of a greenhouse effect to warm the surface. And so people model with other gases in there, and that becomes a question of how much of these other gases could you have. If you're going to say you have methane or hydrogen, you need a lot of it to also produce a greenhouse effect. And again, this kind of ties back to the chemistry, the geochemistry, which is if you were to assume your atmosphere had a lot of these other greenhouse gases in it, you would expect to see some surface chemistry as a result of that, which isn't seen. So this is kind of a paradox right now in the Mars field, particularly ancient Mars field, trying to understand, there's evidence that there should be water, but there's a lot of evidence that, you couldn't have water. And so how do you get this to work? So one possible solution to have water on Mars is collision-induced absorption, or CIA. So what happens here is if you have a gas, and a lot of people model the absorption of the gas as the absorption lines from a single molecule, but if you have a collision between two molecules, for the temporary moment, they form a little super molecule complex where you have two molecules temporarily stuck together in that moment of collision, and that complex itself also has an absorption profile. So if we think of Beer-Lambert's law here, the absorption of a gas mixture, you would have the absorption from one gas, so a cross-section of the absorption of one gas and the density of that one gas, the absorption of a second gas, and a lot of people model those as just individual lines. But if you have this mixture and these collisions, you also have these CIA effects. So you have this squared effect of the density of CO2 squared plus an absorption cross-section that deals with two CO2 atoms together. and you have another gas, you also have that mix effect of density squared of your other gas species and mix, and then you have a mixed term of CO2 colliding with another gas and that absorption cross section. So there was this argument put out a few years ago that maybe we're missing some of the absorption features when we do our radiative modeling, and if we include all the absorption features that are actually present, you would get enough radiative greenhouse effect to trap enough radiation to warm the surface. So the proposals here were mainly based around hydrogen and methane. So if you were to include hydrogen and methane and you include those collision-induced absorption effects, you would get enough trapping. The problem was, at the time, there weren't experimental absorption cross-section measurements of CO2 CH4 CIA and CO2 H2 CIA. So at the time, someone did a proxy measurement using nitrogen to simulate what that might look like, because the nitrogen methane and nitrogen hydrogen CIA profiles were known. And these are these sort of dotted lines here in this figure. And you see that they kind of have an absorption effect in this atmospheric window on Mars. So then someone liked this idea, and they went out and modeled what CO2 methane and CO2 hydrogen would look like, and that is these blue and red lines here. And those are a much stronger effect than the nitrogen one. So this seemed very promising that you include this CIA effect, you would get this increased absorption in this atmospheric window. Of course, the problem here being that these were not experimental lines, these were simulated lines. So this created a need to make these measurements. The other problem with this theory is, as I sort of mentioned, is having high amounts of hydrogen and methane. Now, a lot of models don't really support that, but you could have it as transient warming, meaning maybe you had a lot of volcanic activity for a bit, and that released a lot of hydrogen and methane. Or maybe you had an asteroid collide, and that brought some hydrogen and methane. So for a couple million years, you get a little boost of these gases. You have the presence of the gases now, you have the CIA effect, it warms it for a brief period, you form some liquid water and make some of the geological features before the greenhouse gases dissipate, and then you wait a couple million years for some more transient activity to happen. So this was the current theory. And there was a paper put out in 2015 that supported that if you include the CIA effect, you would get this warming. But again, this is all theoretical absorption lines, and you want real absorption lines. So we saw this and we figured, hey, I'm a spectroscopist. I think I know how to make that measurement. We should do that. So in order to make that measurement, we had to travel to Saskatoon to the Canadian Light Source, the CLS. Now, the reason we went there is because it has an experimental facility that is useful for this measurement. So the CLS, we actually ironically didn't need the synchrotron radiation itself. We just needed to make use of an experimental facility that was at the light source. So we actually got to go during the shutdowns, because they do shut down for regular maintenance. So we actually were able to get a lot of beam time, so to say, because we were able to go when no one else was using it. So it did maybe get a bit of a ghost town while we were there, but we got a lot of work done. So I went with a team of researchers grad students from York and U of T. So this is us out here at the sign. So Carissa from York, Tyler from U of T. And then on a second trip, we went with another York student, Zhang. All right, so what was at the CLS that we needed? Well, what we need to do these spectroscopy measurements is CIA is a fairly weak feature, so you need a very sensitive instrument to detect these weak absorptions. And if we think back to that Beer-Lambert's law a few slides ago, a couple of ways to increase the absorption feature is either you need to increase the amount of gas you have there, or you can increase the path length, the amount of time the light is passing through this gas sample. So a way to do this is something known as a white cell. So it's this figure at the bottom here, where if you have mirrors at both ends of the cell, and you have a beam of light coming in, you can bounce it back and forth between these two mirrors multiple times before it exits the cell. And so for a 2-meter base length cell, you can have multiple bounces, and you can get path lengths up to 100 meters. So the one at the CLS is capable of going to 80 meters, which seemed pretty good. In order to do this detection, you use a Fourier transform spectrometer, which is basically a Michelson interferometer. So a Michelson interferometer, for those who don't know, a beam of light comes in, you split into two parts. One mirror is fixed and it just bounces back. The other mirror moves, which varies the path length, so you produce a time delay when the signal recombines. This produces an interference pattern at your detector, and you do a Fourier transform of that interference pattern, that tells you the power distribution as a function of frequency in your light, which is what you want when you're doing spectroscopy. You want to know how much light is at each wavelength. So this is all here in the far IR beamline of the CLS. So the white cell that I described is this sort of cylinder here underneath. The Fourier transform spectrometer is this box here, and there's a detector at the end path here. So we ended up ultimately being there for a month's worth of time. The facility runs 24 hours, 24/7. So we were doing it in shift work. We had a limited amount of time to try and get a bunch of measurements done. So we had to work really hard for that, but it was a successful trip overall. Okay. So what does that look like when you get an output from a spectrometer? So you run it in two ways. You run an empty cell spectrum first, that gives you your initial light intensity, and then you run it with the gas in the cell, and that's going to give you your final intensity. And then you're looking then for this absorption term here, the difference between your full and empty cells. And so as you do that, you get this blue line here, which is the absorption we measure directly. Now, as I mentioned, this is going to be a combination of several effects. You're going to have CO2 lines, you're going to have methane lines, and you're going to have these collision-induced absorptions. So as you see here, this is sort of now the individual lines do exist in the literature, so we can simulate what those look like. And so you see here, the red line is the simulated CO2 lines, the yellow line is the simulated methane lines, and you see how that matches up with our measured lines. And then we're looking for the CIA effect is kind of a broadband base effect at the bottom of this. Now, unfortunately, these are not the only effects we measured. There is sometimes contamination. We had a lot of water line contamination in the setup, so we also had to deal with water lines. Those are a bit more challenging because we don't know how much water is in there. With methane and CO2 and hydrogen, we know how much of that is in there because we put that in there on purpose. But for water lines, we don't know, so we have to kind of estimate what the concentration of the water is in our setup. There's also baseline fluctuations, by which I mean the intensity of the light source is not fixed with time. It does fluctuate slightly. So there could be slight differences between what your baseline, your empty cell spectra is, and then what the full cell is, not because of the gas, but because of the light power itself changed a bit. So we try and mitigate that a few ways. One way is we do an empty cell measurement before and after full cells. So we kind of sandwich an actual measurement in between empty and full runs. Then we also just look for drift in the signal itself. We know it should be, you can look at places where you know there's zero absorption, and if that doesn't go to zero, you know you're off by a bit, probably because of baseline fluctuations. So there's a lot of sources of error in here, and it's a very difficult measurement to make. All right, but we made it. Okay, so this is the CO2 methane collision-induced absorption measurement, and we did it at 3 different temperatures. The red lines, there are 2 sets. So the dotted red lines is the original prediction from that original paper that was saying that CIA is probably something that would warm Mars. The black line are the measurements that we made. The red solid line is if you add a scaling factor to the dotted line to get it to agree with the black line. And what we find is you need a factor of about half. So the theoretical prediction was ultimately overestimating by a factor of 2. But in general, it was still a good prediction because we found the CIA where it was expected. You see this little absorption feature where they predicted it was. There is a temperature dependence with it that we do see as well. And so basically it was good, it's just it was off by a factor of 2. So this is for methane. I should mention also briefly, the CLS, in addition to having limited time there, we were also limited by the gas concentrations we're allowed. Hydrogen and methane are unfortunately explosive, and so the CLS was not a fan of us making bombs in their basement, so they limited how much methane, how much hydrogen we were allowed to use. So if we think back to just Beer-Lambert's law quickly, if you really want to detect the signal properly, you want to have equal amounts of hydrogen or methane and carbon dioxide in the chamber. We were unfortunately limited. For methane, we were only allowed to go up to 20% mixing ratio of methane. For hydrogen, it was even worse. We were only allowed 8%. So this further pushes our detection limits down a bit. So we're not getting as strong as a signal as we would like to get. Right. Also, there was in the period of between when this paper came out proposing CIA and then we applied for beam time and we got beam time, but before we were able to go to the CLS, another group also had the same idea as we did, and they were able to get beam time a bit sooner than us. And so they were able to make a room temperature measurement, which is this blue line down here. So their measurement came out before we were able to go to the CLS, but they were not able to do temperature dependence, which is something we were able to do. So they had a nice room temperature measurement, which we were able to find agreement with. And again, this sort of, it's half as strong as you want it, as it's half as strong as the prediction, and it agrees with what we sort of see at room temperature. Okay, for hydrogen, the story's there as well. Unfortunately, it's not as clean as a result, partly because, as I said, we can't put as much hydrogen into the setup. Also, the hydrogen had more water contamination than the methane did. So these water lines contaminate, we have to subtract them out, but it's hard to do that, and the water lines kind of wreak havoc in our spectra here. So it's not as good of a result as the methane one, but we still sort of see that, you know, scaling it by half does produce better agreement, although our noise sort of limits us here. But overall, I would call this a successful trip to Saskatoon in the middle of winter, and we got these experimental measurements. All right, so we have the experimental measurements now, and the next question becomes, of course, well, does this warm Mars? So the unfortunate thing here is, as I said, is the prediction was twice as strong as the reality. So it's not going to be as good of an effect as predicted, but you know, maybe it still gets there. So in order to do the Mars modeling, we partnered with a modeler out of Tokyo. His name is Ramsey Ramirez. He was actually one of the original people who did the CIA modeling. He was the one who first used nitrogen before the carbon dioxide prediction was available. So he had this code ready to go. He just needed updated cross section. So we told him, hey, the CIA effect is half as strong as you've been using. If you run it with a half strength, how does that work out? So we went and did that. So this is he did a couple things for us here. He did the surface temperature, the planetary albedo, and then a temperature altitude profile. So for surface temperature, he does a couple different mixing ratios here, comparing to if you were to do CO2 and hydrogen in this case. So if you have up to 10% hydrogen and CO2, you get sort of this best result here. Now the dotted lines are the original predicted CIA values. The solid lines are using the experimental values, so the half strength ones. So we see that with half strength, you're not as strong as before. Before you could get above freezing point, which is this dashed line here going across. You would get, you know, warm atmospheres at much lower pressures, but now you need to go to higher pressures. So the ultimate conclusion here, at least, is if you have even a 5% atmosphere can get to liquid water at under 2 bars, just at 2 bars. For the planetary albedo, this also changes it slightly as well, because you're going to have more warming here. You're going to have more water vapor in the atmosphere, and that'll change your albedo. And then if you look at the altitude temperature profiles, if you have no CIA at all, you're quite cold. If you include CIA, you get fairly warm. And so this is for the best case scenario here. If you were to have 2 bars, I think with 10% hydrogen, you get a nice, pretty warm atmosphere at the surface. So that's promising. For hydrogen or for methane, same figures. The problem here is, unfortunately, you no longer get warm enough at 2 bars. You need to go up to 3 bars in order to get enough warming. This is unfortunately a problem because a lot of atmospheric modeling of ancient Mars suggests that the pressure probably wasn't more than 2 bars. So ultimately, the sort of conclusion here is that hydrogen could still work as a greenhouse gas with CIA, but methane probably is not sufficient. All right, so this is sort of part 1 conclusions on, you know, how do you get liquid water on the surface? So we did the first temperature-dependent experimental measurements of the CIA effects. It agreed in the sense of where it was predicted and the temperature dependence, but it wasn't as strong as predicted. And using these radio transfer models, we show that, you know, methane is not the saving grace we were hoping for, but hydrogen is still in the realm of possibilities. Okay, so that was sort of part 1. Part two is, now assuming that we did have liquid water on Mars and that we have this, would that be habitable? Now, this is reason there's a sort of expression in planetary sciences who are hunting for life. It's just follow the water. Water is really good for life, at least as we understand it on Earth. So if we can find water on other planets, that's a good place to look for life. And if you also think about it, if we're going to keep using terrestrial systems as analogs, if we keep believing that what works for life on Earth probably works elsewhere, if we had a survivable, habitable environment on Earth, it'd probably be habitable elsewhere. This is also important for planetary sciences, and especially space agencies care about this. It's called planetary protection, which is if you found a pretty nice looking planet and you sent a satellite or a rover or surface mission of some sort there, you don't want to accidentally contaminate that planet. So if you find a puddle of water on Mars one day, and you don't want the rover to go dip its arm in necessarily, because what if there was bacteria on the rover that was surviving and now you put it in the puddle and it's able to live and thrive? That's a problem too. So NASA in particular cares a lot about being sure that we're not going to contaminate other planets. So they want to know what habitable environments are out in the galaxy. So for Mars itself, there's a couple things that make survival of bacteria difficult there. It has extreme temperatures, there's very low atmospheric pressure, it's quite dry, there's a lot of UV radiation. Now, the first three aren't so bad. There's cases of that on Earth as well, and cases where bacteria does quite well there. The one that seems to kill all life is the UV radiation still. That still kills bacteria on Earth. There's not many radiation hardy bacteria. So that's kind of the one we're focusing on here is it possible to find a protected environment from UV radiation? Again, with our Mars atmosphere simulations here, we don't assume there's no ozone layer to protect it from UV radiation. So the surface receives quite a lot of UV. So a proposal was out there that, if you had these bodies of water on ancient Mars, could they support life? Would it be possible that the water itself maybe has minerals dissolved in it? So these salty brines could potentially, would that block out UV? And so you could have bacteria living happily just below the surface in these puddles. And that's nice because, as we said, water is good for life. It was protected from UV now. It's probably looking like a pretty nice environment for life. So we wanted to look into this possibility. So this is something we did at York now, was we were just going to measure the absorption of UV radiation through brines. So in the lab at York, we have a UV lamp and a UV detector. So we just use fiber optics to pipe that light through. You have a little cuvette here where you're going to put various salt solutions in there, and we're going to look at the absorption pattern out of that. So this is going to be a simplified version of Beer-Lambert's law than I use for the gas experiments, where we're just looking at the absorption feature of different brines. And now we're looking at a UV range versus before we were using IR. So this work was done primarily by undergrad we had in the lab named Heather Stone. So she made a bunch of salt solutions and measured their absorption properties. So the salts we chose were ones that are composed, were simple salts, but have elements that are found on Mars. So irons, magnesium, manganese, calciums, and then sulfates and chlorides primarily. So this is a bunch of UV absorption cross sections for these various salts. The dotted lines are chlorides, the solid lines are sulfates, that refers to the anion, and then the colors code the cation. So the purple ones are iron 3+, the green line here is an iron 2+, then we have calcium, manganese, magnesium species, and then we have just simple sodium chloride because that was easy to find in the lab already. So what's interesting here is there's orders of magnitude difference between the absorption properties of these salts in the UV. So in particular, at our strongest absorber, we have the iron 3+, and then our weakest ones are sort of magnesium, sodium. What's also interesting is that you can kind of see here that it really depends on the cation is primarily responsible for the absorption. It doesn't matter if I was using sulfates or chlorides as much as it was the cation is what really drives the UV absorption. So if we have this here, we know the absorption profiles of salts. And another thing that's known is, we can estimate the UV radiation at the surface of Mars. You can plug those two together to figure out what would uh, puddle on Mars, how much UV would that absorb. So this is what's simulated here. So we have on the x-axis for all these figures is the depth of the puddle. So we're centimeter square, or so we go from sub-millimeter, 10 to the minus 2. These are all in centimeters, by the way. So sub-millimeter up to about 1 meter scale. And then we look at, concentration of the salts in grams per 100 mils. So what we see here is for these iron 3 compounds that absorb quite strongly, you can get really strong absorption. And so if you have, only like a 5% solution, even at a sub-millimeter scale or millimeter scale, you're almost completely absorbing all the UV arriving at the surface of Mars. If you go to the weaker absorbers, it kind of doesn't matter. Even if you had a really concentrated solution that was really deep, you're barely sort of getting there. So it's unlikely that these are going to provide any protection. But these iron species seem pretty good candidates to protect you. So this is sort of conclusions part 2A. This is the project we did at York here. Summary was that cations are the more important part, and that iron seems to be our best bet, which is great because there's a lot of iron on the surface of Mars. So the next phase of this project was then to actually test this with bacteria. Would bacteria survive in these brines? Now York is not equipped to do bacterial measurements, but there is a colleague of ours at the University of Florida, located at the Kennedy Space Center, who is. So John sent me down to Florida, I guess as compensation for being sent to Saskatoon in the winter. I got to go down to Florida for a bit. So this is actually not a Florida sunrise. That is a Falcon 9 rocket launch I got to see, and that was at 1 in the morning. So it actually does quite a good job of lighting up the sky. If you have the chance to watch a rocket launch, I would highly recommend it. Anyways, so back to the research though. We're going to test if bacteria can survive these brines. So there's two questions now. One is, in the brines, we kind of can tell that they're going to protect you from UV, but the brines themselves may be lethal to bacteria. So we have to first test, can bacteria actually live in these brines? And then we want to test, does this actually work as a whole thing together? If you put UV, you put bacteria in the brines, are they really going to survive? Okay, so we didn't have a lot of time down in Florida. I was there for Teps exchange, the international exchange program. So if you haven't done one, you should do it. This is a great program. And I went down to Florida, and we picked 3 salts from the previous results. So kind of covering the whole range here: magnesium sulfate, manganese sulfate, and iron sulfate. So these are convenient because they all have the same anion, which is good. So that's going to kind of standardize at least that result. And then we have the 3 different cations of 3 very different absorption features. We go back briefly. So we have a strong absorber, a medium absorber, and then a weak absorber. There was 2 bacteria we selected, so Bacillus subtilis and Enterococcus faecalis. So Bacillus subtilis is a spore-forming bacteria, meaning that if conditions are not ideal, it basically hibernates, it reduces its volume, it ceases biological activity, it kind of turns into what you might consider a seed or hibernation. So it's able to survive fairly extreme environments. E. faecalis does not have that ability, so it's a little bit more susceptible to the environment. Now, previous work on these 2 bacteria species have found that they are both capable of surviving Mars conditions. And there's an asterisk there, by which I mean they can survive things except the UV. So they can handle the low pressure environments, they can handle dry environments, they are able to handle temperatures. So that's why these ones were chosen, is they technically can survive a lot of the Mars conditions. It's just there's a couple we don't know. We don't know if they can survive the brines, but we know they can't survive UV. So, I'm a physicist, and I assume most people online here are probably more physical sciences backgrounds, and no one's really a biologist. So I had to learn how to do some biology while I was down there. So the way you do a bacteria measurement is, first thing is you want to prepare your bacteria samples. You want nice, fresh bacteria colonies. So you grow some bacteria on a petri dish. And so you want to find nice, clean little colonies here. You sterilize all your material you're going to need, because you want to make sure you don't going to have other bacteria growing. You only want the bacteria you want to grow. So that was sort of day 1, is you start your bacteria plate. You start this plate the day before, and then the next day you come back and it kind of looks like this, where all the bacteria is grown. So day 1 is sterilize and prepare your bacteria samples. Day 2 is you come in and your bacteria is ready, your sterile equipment's all ready, and so you're going to make salt bacteria solutions. And then we're going to put this in a Mars chamber right now. So this is our Mars chamber here. We have these little test tubes that were filled with bacteria brine solutions. There are these anaerobic packs to take out any oxygen. This is hooked up to a pumping line that's going to control it at a 7 millibar CO2 atmosphere. And we put this thing inside a fridge that's held at 0 Celsius. So we put that in there for some amount of time. And these experiments we were doing 24 hours and then 3-day and 7-day long experiments. After they've been subjected to Mars for whatever amount of time, we take them out. You harvest them where we make a measurement called the most probable number assay. I'll explain that in the next slide. And then once you've done that, you wait a day to get the bacteria to grow to count them. So let me explain how those last 2 steps work. Okay. Now everyone kind of talks about bacteria as, millions of bacteria per milliliter or per surface area. So it makes it hard to count. If you were to imagine you took your bacteria and you put it on a petri dish and you sprayed it all out, you probably have bacteria everywhere and it'd be very difficult to count because all the colonies sort of run together. You kind of look at that like, you can't count individual colonies here. You can kind of count them when it gets thin, but you can't count how many bacteria are there. So what you do is you do repeated dilutions. So if you take your original sample, you put 1 milliliter of your original sample in 9 milliliters of pure water, you've done a factor of 10 dilution. And you kind of keep repeating that. And so you'll keep reducing the amount of bacteria that are available. And at some point, you get bacteria at a countable number. So then you just have to work out, so, okay, let's say you found, after 6 dilutions, you see 20 colonies. You kind of work back, well, so you did it 6 times 10 to the minus 6. You then reverse what you just did, so you have 20 times 10 to the 6 bacteria in your initial sample. And that's how you count bacteria. Now, at least that's how people used to count bacteria. This is a fairly long process to count all these things. So they made a standardized procedure now where you have these well plates. You still do these dilutions, but you put these dilutions into the pair of well plates. And so then you just count how many wells are growing. So these 2 first wells are your initial dilution. Your next 2 sets are a factor of 10 dilute more. These ones are another 10 and 10. So you're looking for where this changes. So at these low dilution factors, where it's very concentrated, all the cells grow bacteria. And at some point, you start seeing, not all the plates grow bacteria. You went from all 100% bacteria growth to only, it looks like, 6 of the well sites grew. And you go down to 1 well site growing and then 0 grow here. So you kind of see this decay curve with dilutions. And there's standardized tables for these well plates where you look up a value of you going from 100% to 6 to 1 to 0. So you look at, it's a code you look up in the table of values of, all right, you had that degree of loss, and then you know what the dilution factor is. You do a multiplication factor to get your bacteria populations. So that's how you do the bacteria measurements now. And so this is why it takes a day, is you spend 1 day preparing this plate, but you have to then wait a day for the bacteria samples to grow. And so you see if bacteria grew in the well salts or not. So that's why it takes an extra day to make your actual measurement. And this is why it's very important that everything be sterilized when you're doing these bacteria measurements, because these only tell you that something grew. They don't tell you that your bacteria grew. So if you don't have sterilized plates, you're going to have random things growing everywhere because you can't tell what bacteria grew. So you make sure everything's clean and sterile. So the only bacteria in here is the bacteria you put in from your samples. And you know that that is your accurate bacteria number. And of course, we do multiple trials of this to get statistics. All right. So that's how you make these measurements. So here are some results. So for the 2 different species looked at, for iron sulfate, after 24 hours in solution, we see these sort of decay curves. So these are some concentrations. We went from 0, so pure water, up to 8% solution. And what we see here is that the subtilist, that's the spore-forming bacteria, it's quite hardy, and it was able to survive at all concentrations for 24 hours, no real loss in population. Ephicalis, however, is a bit more susceptible. It doesn't form a spore, and so it kind of died off fairly quickly. So after 24 hours, if you look at this is a log scale here, so we lost 3 logs worth of the population, and then we didn't even see any at 2% and above. So this is kind of unfortunate because iron was our strongest absorber from a UV standpoint, but it seems to kill some bacteria quite readily. Magnesium sulfate, a little similar story here, where for the iron, or for the Bacillus subtilis, it's able to survive at all concentrations. It's not as lethal to the Ephicalis, but there is still some decrease with increasing concentration. And lastly, the magnesium sulfate. This one actually was quite good. It didn't actually do much damage to either species with increasing concentrations. So that was only 24 hours. So based on that, we decided to go for longer term measurements. So for the long-term measurements, we only took the highest concentration value that had survivability. So for magnesium and manganese, that's 12% mixtures that we tested that both had survivable populations. For the iron, we used 8% for B subtilis for the longer-term measurements, and then only 0.5% for Ephicalis. So for the long-term results, this is where we did 3 days, 7 days, and our initial 1-day measurement. So for iron, after 1 day, that was already sort of seen there for the 1-day measurements, but now we extend it all the way for 3 days and a week long. And here we see that iron, the spores are still able to survive. So iron doesn't seem to have any effect on the spores, but it does keep killing off the Ephicalis as we go longer. So at 3 days, we lost another 2 logs worth, and then we didn't detect any on 7 days. Similar story for manganese. There is still this die-off with longer time, but at least we still have some survivors after 7 days. And then magnesium, again, it's not that lethal, but there wasn't, we didn't notice any lethality at 1 day, but you start seeing a bit of lethality as you go longer. So the question sort of comes then, what is it in the brine that is killing off the Ephicalis bacteria? So we know that B subtilis is a spore former, that's why it's able to survive, but what is killing Ephicalis? So we measured some brine properties. So there's a couple of things we looked at, water activity, electrical conductivity, and pH of the 3 brines we investigated. So water activity, there's no change between any of the 3. They all have this roughly the same water activity, so it's probably not that. Electrical conductivity, manganese was a little, or magnesium is the black line. It's a little bit different than the iron and manganese lines, but again, you want something that's different for all 3 because Ephicalis had a different die-off curve for all 3 species. pH seems to be the answer, where for pH, there's a clear difference between all 3 brines. So it seems to be that Ephicalis is very susceptible to the pH of the solution. So if we want something to survive on Mars, it needs to be able to handle acidic environments, because iron is the best protection from our UV measurements, but also is the most acidic here, and it was one that was causing the most die-off. Okay. So we kind of tested that these bacteria can, some cases, survive in the brines. The last piece of the puzzle is to put it all together and actually test this in a full simulation chamber. So also down in the Florida lab, they have the planetary atmosphere chamber, he likes to call it. So I was working for Andrew Sugar, I should probably mention who the lab supervisor down in Florida was. So this is the planetary atmosphere chamber. It has a temperature control surface plate here where it hooks up to a liquid nitrogen tank, and there's a temperature controller to control set points so you can get nice temperature control for long-term experiments. You can evacuate the whole atmosphere out and then pump in a Mars gas mixture that'll be mostly CO2 with some trace gases. There is a UV lamp here that shines down over the samples. And then if we didn't use it, but if you want to, you can put filters in here to alter the UV flux. So if you wanted to simulate a dusty atmosphere, you can. We didn't need that for this experiment, but other experiments have required that. So inside this chamber, we put little Petri dishes inside with different brines in them. These Petri dishes are 5 centimeter diameter, and so we put 20 mils of water in there, and that gives you roughly a 1 centimeter deep puddle. Now, this is why we wanted to do this actual experiment here, is because the bacteria you kind of assume are going to be uniformly distributed throughout the brine, but the UV absorption is going to be, there's going to be no UV absorption right at the surface, and at the bottom of the Petri dish, you'll have more UV absorption. So even if I had a brine that should absorb UV, there's going to still be some potential die-off because the UV absorption is going to be weak at the top and strong at the bottom of the brine. So we still want to kind of test, does this all work? As another just quick aside, in the back here is the bio hood where you prepare your bacteria samples. It's a lot like a fume hood, but there's also an air vent along the start of it to suck air in. So the atmosphere, the air from the lab doesn't go into the fume hood, but your hands can. So it kind of protects you from atmosphere contaminants in the lab itself. Okay, so this was the final test experiments was in the full Mars simulation chamber. So after just 1 hour of UV exposure in the chamber, these are our results. So control refers to pure water solution, pure water that were also protected from the UV. So when I put them in the chamber, I also covered them with tinfoil so they don't see any UV radiation. They're in the chamber, they're receiving the atmospheric components of low pressure and temperature, but they're not receiving UV light. So that's control. Water is just pure water, they're exposed to UV, and then the iron, manganese, and magnesium measurements. So what we see here is for 1 hour exposure, water doesn't provide much protection. You're losing 3 logs in just 1 hour. Iron holds pretty strongly and making it very similar to the control measurements. Manganese and magnesium are pretty weak. Interestingly, they came off a little weaker than water. Also to note is, again, Ephicalis, because it's more susceptible to the salts, it does always have a more extreme die-off curve compared to the Bacillus subtilis. Now, since iron is kind of our golden boy right now, we focused in on iron measurements, particularly just trying to see is there a concentration where UV exposure drops off, and at least at 1 hour, we didn't see anything significant. So for a bunch of different concentrations, the iron solutions all did very well. This is somewhat expected if you think back to that figure I showed a few slides back of the simulated Mars depths where I was doing different depths and different concentrations for UV absorption. And iron was very strongly absorbing that you didn't need much to absorb completely. Our next step was to move up from 1 hour UV exposures to 24-hour UV exposures. And so here at 24 hours, Ephicalis just straight up dies. So this is all iron sulfates. This is the only one we cared about now. B subtilis, though, you find that, again, at 0.5%, it holds quite strongly relative to the controls. So these iron sulfate solutions are very promising for bacteria habitability. All right, so the conclusions of this last part is the spore-forming bacteria are able to survive in the brines because they're able to handle the acidic environments. But then in terms of actual UV habitability, you want iron sulfate was the only one that was really strong enough to do it. Our other salts were insufficient in providing UV absorption. So I guess the conclusion here is that a brine on Mars is a habitable environment if your bacteria can handle acidic environments. And that is the end of my talk here about water on Mars. I'll take any questions.
I have a question, Paul.
Yes.
On your so a couple of slides back when you were looking at the B subtilis and the other one that I'm sorry, the name kind of as a function of like survivability in the UV as a function of this. Was 8% kind of the highest that you went? I'm just looking at the
Yes, for iron sulfate, that was the highest we went with it. So part of the reason for that is it basically comes back to sort of doing experimental procedure wise is we have to make a mixture of bacteria samples in liquid, and then you combine that in with a brine. So you're going to be diluting your brine a bit. So we couldn't make a really concentrated brine. So we started with concentrated brines that then were diluted when we added the bacteria solution to them. So that's part of the reason why we were limited by how concentrated we can go, because we had to, in this experiment, we were doing half brine, half bacteria solutions. So our concentration actually went down. So I actually made 16% solutions and then added half of that to a water solution. So it goes down to 8%. But iron sulfate was limited at, I think, around 20 is probably the solubility point for iron.
Okay.
For, I think, either manganese or magnesium, one of them is limited by, I think, like 50 grams per mil, or no, 20 grams per mil, or 24 grams per mil. So it's like we had to do double concentration, so we were limited by what the double concentration limit was before we would go down.
Okay. And at the other end of the scale, again, kind of going back to that same plot, you kind of have there's quite a jump, right, from like zero to no, kind of further along the one with the two kind of the 24-hour exposure
Yeah.
ones. Kind of between the 0% and the 5%, there's quite a jump, but I'm guessing again, because you're kind of having to do the dilution, it makes it difficult to get anything
Yeah, so
between
it
those.
was it's also kind of it was more a question of, at the time before we did experiments, we thought, I mean, we kind of thought that 0.5% felt fairly weak. So I think that was kind of a somewhat surprising result on our end, was that 0.5% still actually provided enough coverage. Like when we first started doing this, like it seemed like that felt like a very small amount to mix in. I thought it was going to be insufficient. It was kind of surprising to us that actually it is that strong absorption at that low percentage. So, it would have been maybe worthwhile to have gone back and done lower things, but we kind of already started the experiment doing a high end. So we kind of had to keep going in that route.
Yeah, totally fair. And you're right. Super interesting that 0.5% is sufficient in that case. Thank you.
Okay, I guess that's all the questions then. I guess I'll stop there. Anyone